Welcome everyone to the LRIG Philadelphia Science Webinar. My name is Lorena Kalal and I'm at GlaxoSmithKline in Collegeville, Pennsylvania. I am your WebEx host today. Just a couple of comments about the WebEx. We are recording this for future viewing. Secondly, participants are muted. So for questions at the end, we will be using the chat function. Please send your questions to all panelists, and one of the panelists will read the question to the speaker. With that, I would like to hand it over to the chair of our LRIG Philadelphia chapter. Over to you, Rodney.
Good morning, good afternoon. Next slide, please. Can you switch to the next slide, please? Thank you. Welcome to our virtual scientific meeting. This is our fourth, I believe, virtual scientific meeting where this is an opportunity for scientists to share their research and innovations with the global LRIG community. All our events are free to attend and open to all. You can get information about our upcoming events by going to lrig.org. You'll see a table with various events. We have another event in 2 weeks from today on drug repositioning. So exciting talk that you can go. The link to sign up will be on lrig.org. It's an Eventbrite link, and it's open right now. And on the first Wednesday in April, we have a talk on thermal shift in cellular assays. So that one we don't have registration up for yet. Just wanted to let you know some advanced items. If you're interested in presenting or organizing a future scientific presentation, please write to talks.phl@philly.org with your proposal. A special thanks to Lorena, who's our host today, and we'll introduce the speaker in a moment, Ron Ferguson and Sharon, who are also our committee for these virtual scientific meetings. Without further ado, let me introduce here Lorena, who will introduce our speaker for today.
All right, thank you. It is my pleasure today to introduce Dr. Gyuseok Kim from the Singh Center for Nanotechnology at the University of Pennsylvania. His topic today is nanotechnology and nanofabrication for biological applications. Dr. Kim is a principal scientist at the Singh Center. Prior to joining the Singh Center, he worked at Samsung Electromechanics as a senior engineer on the touchscreen panel and characterized devices and materials with electron microscopy. Welcome, Dr. Kim. Over to you.
Thanks for the introduction, Lorena. Hi, nice to meet you. Today, I'm going to present the nanotechnology and nanofabrications for biological applications. So the presentation consists of introduction and nanofabrication, some key techniques about the nanofabrication, and some biological applications and capabilities at Penn. So let's start with introduction. So before starting the presentation, let's think about how big is nano. So here, 76ers star Ben Simmons is 6 feet 11 inches, so which corresponds to about 2 meters. If we scale up by 1,000 times, the height of the Mount Everest is 9 kilometers. Scale up again by 1,000 times, the diameter of Earth is 12 millimeters. One more scale up, so 10 to the 9th meter. The distance from the Sun to Earth is 150 billion meters. So 150 times 10 to the 9th meters. Let's go back to the other direction. So Ben Simmons is meter scale. If you scale down by 1,000 times, there is a fly. The size of the fly is 7 millimeters. the width of the human hair is about 70 micrometer. So 70 million meter. And if you scale down by 1,000 times, there's a virus. The size of the coronavirus is about 100 nanometer, which is 110 to the negative 9th meters. So we make the device with the nanometer scale. So is this nanotechnology just for fun or something cool advanced research? No. So actually nanotechnologies exist around us. So every day we use cell phones, tablet, and computers. So electronic devices have semiconductor chips. How about as LED and solar cell? These are another semiconductor products. And how about the Oculus? So how does the Oculus detect the motion of the hand? So we need gyro sensor. So the first four applications are or about conventional applications of the nanotechnologies. Here is the fifth one. So recently there's a non-conventional applications. So especially for biological applications. So we're gonna discuss this today. These are the images that these are the images of the devices that was made at Penn. So I'm gonna introduce some devices, biological application devices to you today. And so I cannot introduce all the devices, but I can introduce some of the devices, and you guys will figure out what applications can be applied in this research area, and then what kind of capabilities we can provide at Penn. So let's talk about the nanofabrication, especially the key technologies about first. So this is a silicon wafer, so bare silicon wafer. So most of the nanofabrication start with this bare silicon wafer. And then once the nanofabrication completed, the silicon wafer looked like this. So this is the photo from TSMC. So this is semiconductor. So if you make a cross section of this chip, the chip look like this. Okay. The key is that we started with two-dimensional flat wafer, and this wafer end up having three-dimensional structure with multiple layer structure. How does this happen? So let's think about some simple examples. So for example, this is the left side. So this is a schematic imaging of bare silicon wafer. It's the cross-section imaging. So we started with silicon wafer, and on the right side, as you can see on the right side, we want to make a trench in the silicon. How can we make this? So we have to cover some area with red material. And then we have to do patterning. And then if you remove this red color, and then we're gonna make a trench, as you can see on the right side. So covering some area, the process that covering some area is what we call lithography. And the material we use is photoresist. The process we do patterning is what we call etch. And the process we remove the photoresist is strip. Okay, let's think about another example. As you can see here, we have bare silicon wafer, but silicon dioxide trench sitting on the silicon wafer. So in this case, we have to deposit silicon dioxide first on top of silicon. And then we do lithography with photoresist. And then we do etch to pattern silicon dioxide. And if we remove the photoresist, we can make a silicon dioxide trench sitting on the silicon. Let's think about one more example. So here's a bare silicon wafer, as you can see on the left side. And if you look at the right side, so we want to deposit some thin film electrode. How can we make this? So we have to do lithography first. And we have to deposit the electrode material. And then if we remove the photoresist, we're going to end up having thin film electrode sitting on the silicon. From those three examples, we can conclude nanofabrication process mainly consists of lithography, deposition, and etch process. And one more, lithography is needed for selective opening or protecting of some area. So let's take a look at the lithography, etch, and deposition step by step, a little bit more to understand our applications later. So for the lithography, we have to use the photoresist. In the photoresist, there are two different types of photoresist. One is positive resist, the other one is negative resist. So as you can see on the left side, we have there's a silicon wafer, and we put some positive photoresist. And then once we do exposure, so we expose with UV light, and the exposed area, the chemical reaction occurs in the exposed area. And if we put the sample in the developer, the exposed resist will be cleared. In the case of negative resist, it's reverse. We do exposure with UV light, and then cross-linking happens. And then if you put the sample in the developer, on the exposed area will be stayed on the wafer. So at Penn, we provide some tools with different wavelength of the light. You can use up to 6-inch wafers, and if you are targeting 1 micrometer feature size, you can use mask aligner that uses broadband UV light. If you are targeting 0.4 micrometer feature size or greater, you can use direct write tools. So we use 365 nanometer specific laser wavelength for the exposure. If you're targeting even smaller feature size, you can make up to 20 nanometer feature size with electron beam lithography tool. So next step is etch. So in the case of the etch, there are two, there's one important concept. One is isotropic etch versus anisotropic etch. In the case of isotropic etch, the etch rate of the material is independent to the direction of mother material. For example, as you can see on the left side, if you etch the silicon with xenon difluoride, you're gonna have such an undercut. But if you use some reactive ion edge tool, this is the example of the RIE tool. We will discuss this later. So we can do the etch with some directionality. So we can make a very vertical sidewall by anisotropic etching process. So let me introduce two etch tools, some representative etch tools. One is Reactive Ion Edge. This is one of the most popular etch tool. So in this tool, we use SF6 or CF4 gases. So we use fluorine ion to etch the silicon or silicon dioxide, silicon nitride, or titanium. It's very popular, easy to use, but it gives relatively low etch rate and limited selectivity of the material to the photoresist. The left side of the image on the left side is the example of the silicon reactive ion edge tool. So we etch the silicon with RIE tool and deposited silicon dioxide with PECVD tool, which will be discussed later. Another etch tool is deep reactive ion edge, so-called deep RIE. This is an interesting tool. So the process in this tool consists of two steps. One is passivation, and the other one is etch. First, the tool protects the material with Teflon C4F8, as you can see on the left side of the figure. And then the tool etches the material with SF6. So of course, SF6 etches Teflon a little bit. Okay. Each etch step is an isotropic etch, but the tool repeats this passivation and etch step again and again. And eventually, we will end up having some anisotropic etch profile. This tool provides the fast etch of the silicon. So we use this tool only for the etch of the silicon. Okay, and it gives high selectivity of the silicon to photoresist or silicon dioxide. So next tool is deposition. Next is some tools for the deposition technique. So in the case of deposition, there are two representative techniques. One is physical vapor deposition. The other one is chemical vapor deposition. So let's start with physical vapor deposition. So in the case of physical vapor deposition, we can think about the thermal evaporation first. So everyone knows wafer is vaporized at 100 degrees Celsius at 1 atm. But what if we lower the pressure? We can evaporate the water at 20 degrees Celsius at 17.5 torr. So same thing happens. So if you want to deposit some, for example, gold, copper, so we reduce the pressure of the chamber, and then we add up some thermal energy. Then we can material evaporate, and we can deposit the material on our sample. We can apply the energy with E-beam, not by thermal energy. So this is the E-beam evaporation, as you can see on the figure of the center. If you look at the right side, there's another tool, spotter. In the case of spotter, we use argon, and we mix some plasma. So mechanically, argon plasma hits the sputtering target, like as gold, titanium, copper, and mechanically it hits the plasma hits the sputtering target, and that material is will be deposited on your sample. So we have to use these physical vapor deposition tools for making electrodes. Another important technique is chemical vapor deposition. In this tool, in this technique, we use actual chemical reactions. For example, we can deposit silicon dioxide, silicon nitride, or amorphous silicon with plasma in CVD. Or we can do the diffusion process with silicon dioxide or silicon. We can grow silicon dioxide or silicon nitride with diffusion process, where we can do the atomic layer deposition with this atomic layer deposition tool, or we can do the parylene coating with the parylene coater. So I'm not going to tell you specific tools too much, but if you have a question, I can go back these tools later. Okay. So next step. So some biological applications and capabilities at Penn. So let's think about the scaling. So how does a water strider float on the water? How does this can happen? So this is because they have the nanostructure. So as you can see on the right side of the slide, so let's think about the total area versus the volume. Okay. So if we think about the 1 centimeter by 1 centimeter cubic, the total surface area is 6 centimeters squared. But if we think about the 1 nanometer size cubes, the total surface area increased by 60 million centimeters squared. So at the micro and nanoscale, the gravity and inertia becomes negligible, whereas surface tension, electrostatic force, viscous force, and diffusion become more important. Our body is similar. If you think about the artery, vein, and capillaries, the size of the artery and vein, the sizes of artery and vein are 1 to 10 millimeter, but the size of the capillary is 10 micrometers size. And the white blood cell and the size of the red blood cell is about 7 micrometer. So the capillary, like small scale microfluidic channels like capillary, if the dimension of a cube, let's think about the small scale microfluidic channels like capillaries. If the dimension of a cube is reduced from 1 millimeter to 1 micrometer, the surface to volume ratio increases by a factor of 1,000. The surface force and the surface tension dominant over inertia body force, and the viscosity dominant over inertia. So fluid mixed by diffusion, not by turbulence. So before, some people thought, what if we make a microfluidic devices that has very narrow, small channels with nanotechnologies? So they made lab on a chip concept. Okay? This is what we call lab on a chip. So in the conventional system for biological testing, we need a big, huge area of lab. But what if we make a small device that integrates multiple laboratory techniques that fits on the chip up to a maximum size of a few square centimeter in size? Okay. So these devices were developed recently. So I don't want to say the microfluidic devices are were the best solution, but certainly microfluidic devices have some advantages over conventional technique, because microfluidic devices, with the microfluidic devices, we can work with small volumes. For example, a few tens of nanoliters of reagent or blood. We can reduce the cost of reagent and power consumption. We can increase the speed of reaction. We can almost perfectly control the mixing dosage. So we can do the exact experiment. Can be integrated with other devices. For example, microheaters or some other. If you want to elaborate temperatures, we can put some heaters on the microfluidic devices, and it is easy to use and easy to dispose the device and fluid, minimize the dead space, void volume, and sample carryover, and complex capabilities. We can increase the number of parameters monitored per assay. because we can add up some more channels to put into the devices. So Tsensor is kind of one of the simple examples of the microfluidic channels. So as you can see on the left side of the slide, simple T-shaped we can make a T-shaped channel, and we can flow two fluids. We can observe interdiffusion and chemical reaction of fluids with the optical microscopy, and we can check the diffusion process of two fluids. So this is the example of this Tsensor. The top left side, you can see the geometry of entire device, and the lower right side, you can see some images of the fluid. Actually, this is the DI water colored with food coloring. So one is green, the other one is red. They are immiscible at the first step once they meet each other, but as they flow, the diffusion occurs, and in the outlet they are mixed perfectly. The size of this device is about 1 inch by 2 inches, but you can control. You can make the device as long as you want, almost. So the total length of the mixing channel, so Tsensor, is about more than 50 inches. Okay. Another example is gradient generator. So this video will be a little bit laggy for you, but let's look at this video. So white blood cell is chasing bacteria and engulfing this bacteria eventually. Right here. So how does the white blood cell chase the bacteria? So it's all about the chemical gradient. So we can make a gradient generator with microfluidic microfluidics technique. So this is on the left side. This is the gradient generator that I made with high school students 2 years ago. For example, on the left side, we flow the green fluid and red fluid, and then they flow with some flow rate, and then in the outlet, they make the gradient, as you can see on the lower left side of the slide. So we can use this gradient generator for pharmaceutical applications. For example, you can put once inlet, you can put drug in one inlet and some media in the other side, and then you can flow it, and you can generate some drugs with different concentration, or you can flow two concentrations, some two different flows, and make some gradient, and then in the outlet you can also put some cells in the out. You can make another inlet in outlet area, and you can put some cells, and you can observe how the cell interact with with the fluid with the gradient, as you can see on the right lower side of the slide. How can you make this device? It's kind of it's not difficult at all. So assuming you have never experienced the nanofabrication, but you just come to the Singh Center and there's a TA, teaching assistant, and you start making the device. You can make the device within 3 hours. So this is the process step. So first, of course, we start with the silicon wafer. The first step is dehydration to improve the adhesion in between silicon wafer to the photoresist. And then we have to spin coat the negative resist. Okay, so we have to dispense either SU8 or KMPR, and then soft bake at 100 degrees Celsius and do the exposure. And as you can remember, since we are using the negative resist, the exposed area will be stayed after the development. So before development, we have to do some post-exposure bake to finish up the chemical reaction. And then if you put the sample in the development, any exposed area will be stayed on the wafer. And then this is what we call master. So now master is prepared. Then the next step is to pour PDMS. This PDMS is polydimethylsiloxane. It's kind of silicone rubber. And then we pour this silicone rubber PDMS and cure. And then we cut this PDMS layer and then do some plasma, oxygen plasma to put, to suspend this, our device to the glass. And then characterization. We put some fluids. And this is the final result. And this process takes about 3 hours. So some people, so this the technique itself is pretty simple and straightforward. Some people think about the organ on a chip. This is more new emerging technologies. For example, what if we make an organ on a chip? So previously we discussed the lab on a chip, but now we're talking about the organ on a chip. So this idea brought by some researchers, for example, in the preclinical, so for pharmaceutical applications, especially in the preclinical status. So we use the cells, live cells, we grow the cells, and we put some drugs to see how the drugs affect the live cells. And these cells were grown on the petri dishes. The problem is these cells are not like human body environment. So, our human body is not on the petri dish. So we do have to test with animals, but the animals' cell is different with human cells. So what if we make this organ on a chip? For example, there are 3 channels. So one fluid channel in the center and 2 vacuum channels on the side. And we put a membrane and we put some lung cells. Okay. And there's a capillary cells on the other side. Okay. If you think about these lung cells on the petri dishes, although there's a contraction and expansion during the breathing, actually, we cannot make that kind of environment in the petri dishes. But with the organ on a chip, especially with this channel and vacuum channel, with this fluid channel and vacuum channel, we can apply some contraction and expansion by controlling the vacuum level. So we can give some contraction and expansions to the actual lung cells. And then we flow some air, as you can see on the right side, and we flow some liquid, okay? And we also flow some bacteria, and we also give some medicines. then we can test some actual interaction with the lung cells and the drugs with actual like with some better environment, something like actual human-like. Okay, this research has been done by Dan Hong, Professor Dan Hong at Penn. Another applications of the microfluidics is Droplet Generator. So this is the typical example of Droplet Generator. So we have, as you can see in the imaging, there is a T-shaped device. So we put some dispersed phase through the one inlet, and we also put some continuous phase to the other two inlets, so like as oil and waters. And then if we pinch the dispersed phase, we can make a droplet generator. The droplets are generated by competition between viscous force and surface tensions. The good thing is that we can make a well-controlled in terms of size. So we can make monodisperse droplets. But the problem is if we use the conventional soft lithography technique, like as PDMS on glass, we cannot make a droplets with high speed. So in order to achieve the high throughput droplet generation, we have to think something else. So here at Penn, Dave Isakdor lab and Dayan Lit lab, they made droplet generators with silicon. As you can see on the left side, they put more 10,000 droplet generators on a 4-inch wafers. Okay, and then they did wafer bonding with the glasses and put some continuous phase and dispersed phase, and they extracted the particles. And this in the center of the slide, as you can see, they even made PCL particles with the regular size. So they are all monodispersed. And the size of the this device, as you can see on the right side of the slide, so the size of the device is kind of you can put the device in your pocket of your shirts. And I hope you can see this animation. They were able to make a lot of droplets with the parallelized sequence design. So this is a very good research result in terms of high throughput. They were able to make 277 grams per hour through this method. And this research was published in 2018, and they made even higher throughput device and published another paper in 2019. And this device is very robust because they use silicon-based. They use silicon and glass, not PDMS. And this is highly monodispersed. The coefficient of variation is less than 5%. And we can use this for slow or well-controlled drop release. And the key difference, so now think about the fabrication process. Okay, so they didn't use the conventional fabrication soft lithography fabrication technique. Rather they use deep RIE. As if you remember, if you use deep RIE, you can etch the silicon with very high speed. So they used deep RIE tool actively, and they made big channels from both side and back side, and they did glass bonding, wafer bonding, and they also used excimer laser to make a punch through the inlets and outlets, and they were able to make the this biological this application for biology. So here's another example of biological applications that uses deep RIE. So this is the human blood cell blood filter, whole blood filter that was developed by Wistar Institute and the Singh Center for Nanotechnology. So this is a silicon, basically, and the thickness of this silicon is 100 micrometer, and the diameter of each hole is 9 micrometer. Okay, the key challenge of this process was high aspect ratio, and the process flow was we started with silicon, and we deposited silicon dioxide. We did lithography, and we etched silicon dioxide with RIE tool. And then we etched the silicon all the way through with deep RIE tool. And then if we remove the oxide layer and the photoresist, we have the freestanding silicon device. Okay. And another example is graphene sensor. On the left side, this is the graphene sensor for COVID-19 and recently published. So it was published a few months ago. So the graphene is a very good material in terms of conductivity and sensing capability. So we can use the graphene. You can make a microfluidic devices with graphene. And here at Penn, Professor Charlie Johnson is working on graphene sensor. We also have a lab course here at Penn. So ESC 536, that is, we provide some hands-on processing to the students. So we make the graphene sensor over the course. So we start with bare silicon wafer, and then some lithography, titanium, gold, thin film deposition with the evaporator, and then lift up process, and then graphene transfer process, and then patterning with oxygen plasma, and then if we encapsulate the device with PDMS, we can make a graphene sensor. Another example is newer device. So that has been investigated actively from the Penn Medicine and engineering school. So we can make a electric neural devices with parylene and some electrodes. So this is the example of the ECOG device. So Sharp, the neural device was implanted on the cat brain. And we also have some startups working at the Singh Center for Nanotechnologies. For example, Vigitech, they are making some thin film solutions for glaucoma to reduce the pressure inside the eye. And another example is Inamed. They are making some microfluidic devices for the healthcare from home. So that devices enabled the blood testing at home. So where can you make such a devices? In the States, there is a NNCI national nanotechnology coordinated infrastructure. Across the US, there are 16 universities participating in this program, and they are all supported by NSF. Okay? So usually clean room is kind of closed area because they don't open their facilities to the public. But these 16 universities open their facilities to the public. And Singh Center is one of them. Okay, so Singh Center, we have clean room, scanning proof facilities, electron microscopy facilities. So if you're interested in making any chips, any devices for any applications, please come visit us. We welcome you. Thank you.
All right. Thank you very much. So we will take questions. Dr. Kim, can you advance to the next slide so that I can just make sure that people know how to give us the questions, right? So you will find in your chat box, if you select all panelists, I'll take a look at some questions. So we have one for you. What is the long-term stability of nanodroplets, and have nanodroplets been used for sustained release of drugs over an extended time?
Well, to be honest, I don't know because this is kind of newly developed technique, and so I heard that the first author, Sagar is my friend. He told me this was collaborated with GSK. Maybe you know, this nanodroplets can be tested in the GSK even more, but I don't have the number how long it can be sustained.
Okay, thank you. There's another question. Can you explain how the graphene sensor detects COVID-19 antigen? Sorry, are you frozen? Oh, you're going back, yeah.
Yeah, I'm going back.
Yep.
So yeah, this is example. So well, I don't know the exact detection mechanism. Maybe some people in the biological area can explain some better. The key in this device is graphene. Graphene has very low conductivity, so and it's basically carbon, right? Carbon with some free electrons floating. So graphene captures the COVID virus or some materials really well. So its sense is really, the noise to signal ratio is really great, and it can detect very high accurate result compared to any other materials.
Okay. Next question. Would there be any device to screen or sieve subvisible particles in biotherapeutic products?
Well, that's a good question. Let me think
maybe
about
the
it.
sieve relates to that example where you showed the separation of the cells, different sizes.
So yeah, in my memory, so there was a device. For example, there's a, do you see me? There's a channel. There's a channel with multiple posts. Okay, and then the post is located, they are placed with the diagonal directions. If it flow from one side of the channel to the other side, and then the particle should pass through the post. Okay, so small particles, they can go through the post really easily, but the larger particles have to go around the post. So we can you know, we can split the particles with different sizes and observe that. Okay.
Yep. Thank you. Do one last one before we wrap up. So this technology seems to have found excellent applications in biology and chemistry. Do you have any insights in what will be the next cutting edge applications in both? Anything beyond tissue on a chip or lab on a chip?
Yeah, that's a great question. The organ on the chip is recent, very hot, cutting edge area, I think. I think personally, I think we can even make the devices that can be implanted inside the body. For example, you know, we can put microfluidic chips in the body that to control or sense some biosignals. Okay. The next thing could be implantation and hold that.
Yep. All right. Well, I think it's time. We're at the time. So I'd like to wrap up and thank you very much for Dr. Kim for your presentation. And I'll hand it over to Rodney to wrap up. Thank you again.
Welcome.
Thank you for your excellent talk. I'd like to thank everyone who attended and encourage you to register for our meeting in 2 weeks from today on drug repositioning. So have a fine afternoon. Thank you again.